Hey everyone, and welcome to this explainer. Today, we're diving headfirst into the complex, but honestly absolutely fascinating world of genotyping. Think of DNA as the ultimate instruction manual for any living organism. Today, we're not just going to talk about how to read that manual, we're going to explore exactly how scientists pinpoint those tiny, single-letter variations in the text that make every single organism completely unique.
So, here's our roadmap for the day. We'll start with decoding the genetic blueprint, check out the types of genetic variation, look at how genotyping actually works, explore some real-world applications, and finally dive into the solutions provided by CD Genomics. Let's get right into it.
All right, Section 1, Decoding the Genetic Blueprint. We're going to start by zooming all the way out just to get a solid grip on the foundational concepts before we hit the heavy machinery. To kick things off, we really have to ask the most fundamental question here. What exactly are scientists doing when they genotype an organism? I know it sounds like a bit of a buzzword, but it's actually a highly targeted, very specific scientific process. Basically, genotyping is the process of reading through the genetic manuscript to hunt down specific variations. You're taking a DNA sequence and comparing it against another sample, or maybe a reference sequence. The whole goal is to identify particular alleles, the alternate forms of a gene, at specific locations. And this is the absolute crucial step that lets researchers link a specific genotype to a physical, observable trait.
And this number brilliantly illustrates the sheer, mind-boggling scale we're dealing with. If you look at a standard plant genome, like corn for example, you're going to find roughly 3 million of these single-letter variations just scattered throughout the genetic code. They pop up about once every 1,000 base pairs. So when researchers are genotyping, they aren't just looking for a needle in a haystack. They're looking for a microscopic needle in a massive haystack.
Moving on to Section 2, Types of Genetic Variation. Now that we grasp the monumental scale of the genome, let's talk about the specific types of typos we're actively searching for.
The really crucial thing here is understanding the difference between the two main types of genetic markers. On one hand, you've got SNPs, or single nucleotide polymorphisms. These are literally just single-letter swaps in the DNA base, like an A swapping out for a G. On the other hand, we have indels. That stands for insertions and deletions, and it's exactly what it sounds like, entire missing or added chunks of genetic text. Both of these are massive drivers of genetic diversity.
Now, what's really wild about this is that when SNPs happen, they aren't just random, chaotic mistakes. Statistically, there's actually a pattern. Theoretically, a single base could mutate into any of the other 3 bases, right? But in practice, transitions, which is when a purine swaps for purine or a pyrimidine for a pyrimidine, outnumber transversions 2 to 1. They frequently appear in specific CG sequences, often because of chemical processes like methylation. It is just fascinating how even genetic mutations follow mathematical rules.
Section 3, How Genotyping Actually Works. Let's see how all this biological theory translates into the actual mechanical toolkits scientists use in the lab.
You see, there isn't just one single way to read the genetic manual. Researchers have an entire arsenal at their disposal, and it's all about picking the right tool for the job. Need a really quick, targeted check? PCR-based methods like KASP have you covered. Looking to screen thousands of known markers all at once? Microarrays are your best friend. Then you've got MassARRAY, which relies on incredibly precise mass spectrometry. And for the ultimate wide-angle view, they're the heavy hitters, next-generation sequencing platforms like GBS and 2B-RAD, which give you massively parallel data generation. Choosing the right tool is always a delicate balancing act. As you can see here, DNA microarrays offer incredible throughput and versatility, but they do rely on specific pre-built platforms. TaqMan fluorescent probes are super accurate and easy to interpret, but hey, synthesizing those custom probes takes time. And while direct sequencing is definitely the most intuitive method because it can discover brand new, totally unknown mutations, it has historically been held back by higher costs and lower throughput. Although, to be fair, new tech is changing that really fast. I really want to highlight one super innovative method here, MassARRAY. If you need speed and precision, but you want to dodge the cost of fluorescent probes, MassARRAY is absolute genius. It uses MALDI-TOF mass spectrometry to literally weigh the DNA bases. Because different bases have different physical masses, the machine spots the mass disparity at specific SNP sites, working within a range of 4,500 to 9,000 Daltons. It even allows up to 40 multiplexes in a single tube and turns around results in an astonishing 8 to 10 hours. No way, right?
All right, Section 4, Real-World Genotyping Applications. We know what it is, and we know how the machines work, but why does reading millions of DNA letters actually matter in the real world?
The impact here is literally world-changing, especially when we talk about agricultural research. By integrating these microscopic molecular variations with observable data, agricultural scientists aren't just guessing anymore when they breed plants. They can essentially molecularly design high-yielding, stress-tolerant crops. We're talking about precisely predicting traits like drought resistance or overall crop quality. This accelerates precision breeding programs and unravels complex population genetics like never before. It is all about designing a much more resilient future for our global food supply.
And that brings us to Section 5, Solutions by CD Genomics. With millions of potential markers and massive amounts of data, who can researchers actually trust to handle their genotyping pipelines?
Well, this is exactly where CD Genomics steps in. As they highlight right on their platform, they have over a decade of focused experience, making them a highly trusted, premium choice in the industry. Navigating these kind of complex genetic data takes way more than just fancy machines. It requires deep bioinformatic expertise and serious scalable capacity. And that is exactly what they've built their reputation on. Let's take a look at their specific toolkit. Whether you're conducting broad whole genome SNP screening using Illumina microarrays or genotyping-by-sequencing, or maybe you need highly targeted fine mapping with MassARRAY or ABI TACMan, CD Genomics literally has the exact scalable platform you need. They even offer copy number variation and microsatellite services, and the versatility is incredible, whether you're working with animal, plant, or microbial genomes.
Now, for the researchers tuning in, I know the practical details matter. CD Genomics has heavily optimized their input parameters to make your life a whole lot easier. They accept standard genomic DNA across various services with really accessible quantity thresholds. For example, their highly reproducible 2B rad sequencing method requires a recommended quantity of just 200 nanograms of DNA at a minimum concentration. That makes it incredibly resilient even for projects where your sample material is super limited. Let's look at how this all comes together. Working with CD Genomics is a true end-to-end partnership. They don't just run your samples through a machine and call it a day. They start right at the experimental design phase, helping you select the best markers and platforms. Then comes the execution, featuring rigorous quality control. And finally, they provide comprehensive bioinformatics, handing over raw data, rich annotations, and linkage disequilibrium results, all cleanly formatted and totally ready for your analysis.
So, the Genetic Instruction Manual of Life holds the answers to biodiversity, agricultural resilience, and evolution itself. But remember, your data is really only as good as the tools you use to read it. With the advanced platforms of CD Genomics at your disposal, what microscopic typo are you going to discover that ends up changing the world? Thanks so much for joining me on this explainer, and keep exploring.